So first, thanks to the audience and to the organizers for letting me present. This is about a botanical drug candidate we call W17P47, which can be seen as an intracellular SARS-CoV-2 growth mitigator. I'll go quickly and briefly through the science behind it, the discovery process that we made, the animal model validation, and the target indication we are thinking of, and the next steps we needed for funding. So this is a slide a little bit complex, but is to say that we have biophysical background, medicine concepts, and on network polypharmacology. Really what we are thinking of is to see the difference of robustness between normal cells, for instance, and cells that have been dysregulated, as it has been shown in the literature for cells infected by viruses. We are reducing this to practice by using mixes of edible compounds. This has been described on a theoretical point of view in a publication, really where we show that when you expose yourself to massive diverse mix of chemicals, which is what is done when we eat food, we submit our bacterial and cellular systems to massive perturbations. Most of this is, of course, without any effect. That's why we can still eat food. But you can show that there could be some that can be globally deleterious to less robust systems, such as microorganisms and cells infected by a virus. So what we showed then is that there's an edible plant paradox. They are safe despite being mixes of hundreds of actives, and the authorities also recognize this as safe, as they encourage us of eating mixes of edibles. So the toxicity issue is totally over. But we know that there are actives in plants, in botanicals, that have activity against bacteria, against cancer cell lines, and at least from what we show here, against viruses. So when we are eating food, we are performing massively parallel phytopharmacology, which is naturally safe. We reduce this in practice in making water extracts of complex mixes of edible plants. We call them wet maps. So we're using water extracts because this, for once, allows to make sure that our compounds are water soluble by definition, and also it reduces any additional risks of toxicities, as can be seen with essential oils that may accumulate in adipose tissues and become toxic. The other interesting thing to note is that because we can show the mixes are synergistic, that makes we create patentable actives. There's a technical issue, however. From a library of as little as a few hundred edibles, there is an enormous number of possibilities to be done. And so a traditional brute force screening or discovery is not an option. We are able to tackle this as a small startup because we are using specialized artificial intelligence algorithms that really allows us to do this. And I'm going to show how it works on that example. So we decided at some point, among other projects, to anti-SARS-CoV-2 discovery. It also documents here how the technology works. We start initially with defining an assay. Here it's growing Vero E6 cells and inoculate them with viruses and observe the viability of the cells. And then we submit this to mixes, and we start first with randomly assembled mixes. This is on purpose because we do not want to use any prior knowledge on the potential activities of the botanicals. For once, because very little is known, and so we prefer to make the hypothesis that we do not know anything. And also we want to be able to patent this later, so we want to maximize the chances of discovering something new. So in that graph below, the gray bars are the effects, the survival effects we observed with the randomly assembled mixes. Each bar represents the result for one experiment. There's not much activity, that's good for us. So we enter all this quantitative data in our algorithms, and they propose us potentially improved mixes. We assay them again, and these are the light blue bars, and we see that we somehow improved already the activities. Then we enter data into the algorithms, and they propose us still improved mixes with the darker blue bars and then the green bars, and then we stopped with the red bars with the final optimization. So what we did next is to check our best candidate, which was W70P47, the one with the most activity, the most potent one, and we checked all different coronavirus variants that we could do with our CRO who was making the experiments, and in reference to the Wuhan or the strain. So actually we see that our extracts work at least as good on any variant, whereas we looked also for precision medicine compound, which had a lot of publicity, but turned out not to work on all variants, especially on the USAWA1, where there was no activity. We then made dose response experiments and to find an optimal concentration, which is our target concentration for in vivo, and which is high enough so that we can think of administrating this to animals and eventually to humans. So we used then an animal model, and the animal model that we choose is the mouse model, which is a lethal model, which is a very severe model and which has been little used because of that. But initially, the authors of that article, they tested it and showed that all mice die, but if they administrate serum from convalescent human patients, so that's some sort of an antibody therapy injected, there is 25% survival. We also choose that model because it's based on a transgenic mice that expresses the ACE receptor in the whole body. And though this was highly controversial 1 year ago, now I think everybody may agree that COVID, the coronavirus, is expressing and developing nearly everywhere in the organism, which explains then the also the long COVID problems. The results for the animal model are shown here. So to the left, we have a Kaplan-Meier survival curve where we see that all the mice untreated die, and the treated ones show 30% survival. So here the WEC map is administered by ingestion per ROS. So it's not intravenous like for the patient serum. So the survivors here, the 30% are real survivors, and we had other results that also make sure of this. But we see it also on the morbidity curve, where we see that for the untreated mice, they get worse and worse until they die. Whereas the treated mice, they end up recovering. We see it also on the weight curves, where the untreated mice lose weight until they die or are euthanized, and the treated mice, then they recover the original weight and are getting well. So we see that with our WEC map, we get a survival which is equivalent to what you get with injected convalescent parent serum. And again, I stress it, this is by oral administration. So as we could here demonstrate systemic efficacy, we're thinking now of going into a clinical study in humans. So the next steps what we need to do is, one is to decide on which indication to go. There are two options, acute COVID or long COVID. Acute COVID, we believe it could be difficult to compete with small molecules, and for a number of reasons, and allow me not to comment on some of them here. And also one can think that could be a reduced market because it may eventually be administered only to acute cases. Long COVID is an unmet need, can be seen as an orphan disease. There could be much easier to convince the doctors to evaluate the product because there is obviously no reduction in chance of treating these patients with an experimental medicine. And the market may be actually quite large, because up to between 5 to 20% of COVID survivors complain about long COVID symptoms. We have then to work also on the formulation options, could be dry powder or liquid glycerol syrup. We find also production partners and validate them to make the product. One thing we see as all, there are preliminary steps before filing the IND or going into clinics. We could extend the optimization with our AI-guided discovery, because this would with higher potency, we could really think of tablets, and also it would mean reduced production costs, as you would need less extract per treatment. Is to say on the experiments we did them from November 2020 to February 2021, and in February 2021 the vaccines were coming out, and everybody was claiming that by May or June one would not talk anymore about COVID. So we wanted to go quickly in the animals, and we stopped optimizing. It should be possible to do better. If we think it's also worth documenting the mode of action. There's of course that systems biology mode of action, which is understand on a statistical and theoretical point of view, but we have also preliminary results through a collaboration with a French university hospital, who are measuring cytokines and to look on how the inflammatory pathways Th1 and Th2 are triggered, and seems to be quite favorable from what we see. And then we think we would be ready to file an IND for long COVID clinical phase 2 in the USA as a botanical drug. We believe we can go directly into phase 2, because all the botanicals composing W17P47 are all considered as GRAS. They are on the positive list of European Union botanicals allowed for food supplements, and all the production process is a food grade process. So in terms of technical methods, it should be accepted as safe without any problem. For funding to go further, we think we need up to 1 million to go up to the IND with all this in the best conditions, and up to phase. This has to be refined because they could, depending on the option and the number of questions, but we would estimate it to be between $5 and $10 million, and we see this in a two-step funding as the ideal. Some part of this will be used also to increase the staffing of our company, which is presently quite small, and we would need especially to hire project managers for clinical issues, production, and maybe botanical. But with this, the time to IND could be quite small, short, depending on the means we have, and between somewhere between 6 and 20 months, 18 months, sorry. So if I can go to the next slide. Why? Yeah, it's just to go through the current team in the company, which is myself as a co-founder and CEO and scientific guy. In the operations, we have also Clemence Marinas, who is a kind of operation manager or kind of COO. And we have 3 full-time technicians. We are backed also for advice and for business development and funding work by Daniel Valtuena, who is a co-founder of the company, Tom Bell, who is acting for the business development, and Jacques Chevalet, who is a former executive on botanical products in. And Alpha Nosos is in fact a commercial phase deep green tech health tech based in France. Our business model is really to be on the discovery side and to license our IP to large commercial and industrial players. We have already patented some of our discoveries internationally, and with patents issued on the national level in Israel and Japan and 2 other countries. We are working mostly now on self-care and cosmetic projects, on discovery partnerships with multinational companies, including really world-class, for 2 of them, multinational companies. We have also some self-care products that are put on the market to be demonstrators of best and also of customer feedback to demonstrate the efficacy. And we are currently funded by business angels and by public loans. We are all, as an exit, we are all interested in either going public or being acquired by any large company who would get enough interest in us. So thank you. This was, hope I was short enough, and thank you for your attention. And of course, I'm very happy to answer any question today or later through other correspondence.
Thank you, Pascal. It's a very important product in the era of pandemic. And, you know, those preclinical studies are very important to make people trust, right, how the product works, and you know, to build on that. And in the US, there's a fast track to develop COVID product through the clinical development phase. Have you considered that or thinking about that?
No, we don't have enough funding presently to go further on the COVID product alone. We hope to have to trigger some partnerships by that time, but that did not happen. I think we're not the only one, from what I can see in the news and in the. It's not an easy field. It's really a field where you need somebody who wants to run that fight. I mean, to get a non-Pfizer product further on the market, which I think is an issue in some instance. So we are putting our attention to the partnerships that we have in place, which are on antimicrobial applications, for which we have already very good results. But of course, we see and we believe in the value of what we have discovered and could still improve, and that seems to work eventually. And well, what we are missing is enough funding to go ahead on that project.
I understand.
Yeah, I have a question. David Allen in San Diego. What is the composition of your product? And you're using AI to identify the actives. But I saw at first you had, what, 5 to 9 servings of fruits and vegetables up there on your screen. So can you tell me more about your composition?
Yeah, the whole idea of what we are doing is to bet that even that we can use safe botanicals in order to, by combining them in mixes, to get activity. So all the botanicals, I don't know them by heart, are botanicals that are on the positive list of food supplements for Europe. The exact composition, what we know is that, well, we know the composition, of course, but there are 2 that are of interest. One is Siberian ginseng, and the other one, sorry, it's a bit special for me here. They can carry claims that they contribute to improving your natural defenses. Doesn't mean much, of course, but these are the kinds of botanicals. Among them, there's green tea, it's not a big surprise. There have been data in the literature showing that green tea could interfere with COVID development in vitro. But there are a few others also which are not known for any effect in it. And the composition has also antimicrobial activity, and so it's protected by composition by our first patent.
Thank you.
Excellent, excellent work.
Steve,
Yes.
let me read a question from the chat, then I will let you.
Okay.
Okay. So Pascal, the acute COVID and the long COVID may have a different pathologies. How do you move forward on this?
Yeah, you are right. We cannot. We are betting on the fact that though the long COVID could be of many different causes, that is still due to cryptic expression of a virus. If this is not true, we might not have any activity, despite the fact that we have positive effects on the inflammatory response of patients. But this would be another mode of action. Here, what we see is that from the in vitro, it seems that we hinder at least and reduce the development of a virus, so we reduce the expression of the virus. So certainly we reduce the inflammation, the part of it that it may have, and if the symptoms are due to cryptic expression of a virus, we might have an effect. If it is not, we may not. So this is it can be of disputable, but I'm not sure anybody has really the response. And from some papers are going towards the direction that there's still the virus is still developing at a very low level, which could explain the symptoms.
Thank you. Thank you very much. Steve?
Yes, I think that's a very interesting application of AI. You often hear of AI being used even on the high throughput screening, but not necessarily on botanical application and the network medicine. And so I think that's a very interesting application. And also, when you look at long COVID, now here in the United States, the US government seems to be investing a sizable amount of money into studying long COVID. And I'm not clear on exactly how they're going about doing so, but they've allocated some $1.4 billion, and have also already activated one program here called Recover that looks at the long-term implications of COVID, and that they funded that program at some $480 million. So I'm not sure exactly how those programs are going to be evaluating therapeutics, if at all. But it's very curious. We are exploring how money is being invested in that domestically, and maybe somehow, because this product is an assortment of food products, food ingredients, somehow it may be able to make it into some of those evaluations. So it'd be curious to follow. Roger, we'll follow up with him and look more closely at this.
Thank you, Steve. Absolutely. Those are great products that we'll definitely follow up on. Thank you.
Thank you.
Any other questions? Okay, then thank you, Pascal. And we're so glad to learn the advancement in the COVID area, which is everyone pay attention to, and we'll definitely get in touch. Thank you for your time. Take care.
Thank you. Bye-bye.
Thank you.